So, what are these tumor antigens? We know that antigens are those foreign substances that are capable of stimulating an immune response against them, particularly production of antibodies. Similarly, tumor antigens are those molecules that are produced by tumor cells, and they can potentially be recognized by the immune system. Many tumor cells produce antigens, which may be released in bloodstream or remain on cell surface, and they have been found in many carcinomas, lymphomas, and many solid cell cancers. Here, a key role of the immune system is to detect these antigens, so to permit subsequent targeting for eradication. There are a number of different types of tumor antigen, including viral proteins. The viral proteins that normally act as antigen for themselves can also act as tumor antigen if that invading virus is capable of tumor formation. For example, Epstein-Barr virus, EBV, and human papillomavirus produce specific proteins such as HPV protein, E6, and E7 in cervical cancer cells. These proteins then act as tumor antigens and help in tumor detection, that are also called as oncoviral proteins. Next category is developmental proteins or oncofetal proteins. These proteins are normally expressed during development process or during fetal life only, but are absent in normal developed adult cells. However, in case of tumor, some oncofetal or developmental proteins re-express themselves on tumor cells. For example, carcinoembryonic antigen, sqadia, is normally expressed on many tissues during fetal life, but absent in adult life. But in gastrointestinal cancer, sqadia can be abnormally expressed on tumor cells that can generate immune response against them, thus acting as tumor antigen. Next one is lineage-specific proteins. These proteins are not only expressed in cancer cells, but also in the normal tissues from which those cancer cells arise. For example, melanoma is a skin cancer of melanocytes. Normal melanocytes and cancerous melanoma cells both express the enzyme tyrosinase, which is not expressed in any other cells, normal or abnormal. So these lineage-specific antigens are present in either normal or abnormal cells of same lineage, and thus they are detected as tumor antigen only based on their intensity. Now the last category of tumor antigen is proteins produced through translocations. As we have already discussed in lymphoid tumors, that one of the reasons of tumor formation is chromosomal translocation. That alters gene positions over chromosomes. So proteins that are produced through these translocations are those that are present only on tumor cells, and thus can be detected and can generate immune responses. For example, as discussed, under chromosomal translocation, the BCR-ABL fusion protein that is formed as a result of BCR-ABL1 translocation is an oncoprotein that stimulates uncontrolled cell proliferation, and also it can be detected as tumor antigen. Ideally, a tumor antigen should be present or expressed only by tumor cells and not by normal cells in order to be recognized as foreign by the immune system. In reality, most tumor antigens are overexpressed in tumor cells, but in small amounts they are also expressed in normal cells. Like developmental and lineage-specific proteins are poorly immunogenic because they are expressed on both in tumor cells and normal tissues as well. But viral and fusion proteins tend to be more immunogenic because they are never present in the normal individual, but only found in tumor cells. So tumor antigens are expressed in two ways. Either they are tumor-specific antigens or tumor-associated antigens. Tumor-specific antigens, TSAs, are found in cancer cells only and never in healthy cells. For example, point mutations in tumor suppressor gene P53 can lead to the development of tumor-specific peptides that do not occur in normal cells. And tumor-associated antigens, TAAs, are those that are found in both healthy and cancerous cells. But in cancerous cells, elevated levels of these antigens are expressed, and in normal cells, they are expressed in small amounts. For example, alpha-fetoprotein are present in normal fetal cells, but are overly expressed in adult patients with tumor of the liver. So this was all about the introduction of tumor antigen. In the last few years, immunologists have put much effort in finding different types of tumor antigens, as they can be the main target for immunotherapy against cancer. If tumors do express antigens that can be recognized by the immune system, the big question is, how do cancer cells avoid destruction and develop into full-blown tumors? Well, we will answer this question in our next section.